What are nucleotides? Nucleotides are considered the building blocks of DNA and RNA. They consist of a sugar, a phosphate, and one of four complementary bases. These four bases are adenine, thymine, guanine, and cytosine. What are Okazaki fragments? Well, first off, why are you asking me? Ask Reiji Okazaki, who discovered them in Japan. In the leading strand of DNA, DNA synthesis is continuous, while on the other hand, in the lagging strand of DNA, DNA synthesis is discontinuous, resulting in fragments across the lagging strand of DNA. Now, where does DNA replication occur in the cell cycle? DNA replication occurs in the cell division part of the cell cycle. Now, what is DNA replication? DNA replication is where one complete strand of DNA is copied, therefore resulting in two identical, completely same copies of DNA. It can be commonly known as hitting an invisible copy button, which is like creating the same of two cars, two books, or two Legos, or stream of two Donald Trumps. Wait. No, we do not. We already had Hitler. No, we don't need twinsies. Goodbye, no. Now that you know where DNA replicates in the cell cycle, and much less what DNA replication is, the real question now is how. And no, not how I met your mother, but how does DNA replicate? What causes it to replicate? Well, this is caused by 6 enzymes. DNA polymerase 3, which adds bases, DNA polymerase 1, which removes RNA primers, DNA ligase, which is the glue that prevents everything from flinging apart from each other, helicase, which untwists DNA, primase, which adds RNA primers, RNA primers are pretty much support beams which keep everything together, and telomerase, which adds a cap to the DNA once everything is therefore completed. These 6 enzymes are crucial to the successful copying of DNA. What are the steps? DNA replication starts in an area known as the origin of replication, also commonly referred to as point of origin. This is very important, crucial, and vital to understand, as DNA replication does not start at the ends, but rather somewhere in the middle. Helicase is the enzyme responsible for untwisting the DNA. This results in an untwisted version of DNA looking like a Y. Then primase comes along and places down RNA primers according to the leading or lagging strands of DNA. As shown from the picture right now, on the leading strand of DNA, an RNA primer is placed at the end of it, serving as the starting position for DNA synthesis, while at the lagging strands where the Okazaki fragments are found, RNA primers are placed all throughout to serve as the support beams. Then, DNA polymerase 3 comes along and places down complementary bases next to these RNA primers. Then, DNA polymerase 1 comes around and removes RNA primers and replaces it with DNA, as well as telomerase comes around and adds a cap to the end of the DNA. Finally, DNA ligase is added to keep everything together and keep everything sound, structured, and safe. Ta-da! You did it! Now you know what DNA replication is, where it occurs, and all that good stuff. And as always, don't forget to like, comment, and subscribe for more content. Thank you! Goodbye!